Well, welcome. My name is Sylvain Coste. I'm the project manager for Open Science in Space Biology, and today I'm going to talk about our division, which is a new comer in SMD. It's the Biological and Physical Science Division, and we use spaceflight environment to study phenomena in ways that cannot be done on Earth. BPS research is focused on two priorities: quantum science and space biology investigation that enable us to strive in deep space, and we call that TIDES, T-I-D-E-S. Our work is done primarily on the International Space Station, where we fly all kinds of different experiments. So without further ado, let's dive into the ISS, and we're going to see a couple of things that we work on. So as we zoom in, you're going to see different plants being grown on the International Space Station. We look at microbe, yeast, cucumber, different plant. The mouse is a big one. And then we also do cell cultures. So we can see here some cell culture being done. We'll walk you through more. This is Jessica Meyer. She's one of our astronauts changing some media for cells. They do a lot of fluidics in space. This is Carla Barron, who's taking picture of the seedlings here as they're growing them. And then this is actually an incubator where we can put the cells into the incubator and grow them for quite a while. So before I start to talk about open science, I'm going to talk about our division, the Biological and Physical Science Division. And we're going to start with the different experiments that we do in the space station, starting with rodents in space. So those missions are called rodent research. And then as we zoom in those, what we try to study here is the impact of spaceflight on mammals. So they have, we track the CO2 level, the carbon dioxide level, the temperature, the humidity, the light cycle is affected for these animals. And of course there is no gravity, so they float around, as you can tell. And this is a good model to understand what happens to our astronauts as well. One thing here that you see is April Ronca, which is a PI in NASA Ames Research Center, discovered this movement that the animals actually adapt to space by starting to circulating in the cage. And they end up having some response from this work and developing more muscle mass than expected from microgravity. So mammals is one. We also do a lot of work on plants in the Biological and Physical Science Division. So let's zoom. This is Megan MacArthur, who is actually going to show us some growth of chilies in space. Chilies are a good thing to have because astronauts lose the sense of taste in space, and so they love having spicy food. So they're very excited about this, and we take time lapse of these chilies. There's actually some science being done here as we study all these different plants being grown on the space station and trying to optimize in a closed loop system how you grow plants in the most efficient way, and that can really have application on Earth. So those are radishes being grown on the space station. All these time lapse are being analyzed, and sometimes the plants are being eaten. Next is we can do sequencing in space. This is kind of a big deal. This is Kate Rubin. She is the first person in the world to do DNA sequencing on the space station. She's using a device called the Mean Ion. This is actually a nice view of the Mean Ion here with the cupola in the background. This device has been used by many to do sequencing on the field, and that's been used on the space station as well to do the first bacterial sequencing, and that was extremely successful. This is actually Kate talking to the Mean Ion team on the ground to troubleshoot some work. Next, we're going to look at what do you sequence microbes. Where do you get the microbes? Well, you get them on the space station. So this is actually Megan MacArthur again doing swab of the walls. They collect the swab across the station. They find the location, and they put these samples into tubes that goes back to Earth for analysis to understand what kind of microbes thrive in space. Let me move there. So we also use animal models, like small animals, like the C. elegans is used for understanding the impact of this is a good model for muscle for humans, and we can compare microgravity with centrifuge data, which is the 1G control on the space station. We do the same thing with Drosophila. They are typically used for neural behavior. And then here you can see actually there are little modules inside the MVP. It's a hardware we use, where we also have 1G control. So all this work is being done. Now, space radiation is a big deal, but if you want to get good space radiation, you go to Brookhaven National Lab. NASA has a partner with DOE, and Brookhaven National Lab has the RIG, which is the Relativistic Heavy Ion Collider. And we have our extraction line here called the NSRL, the NASA Space Research Radiation Lab. That's Adam Rusek is the boss over there, and he's basically, this is your beamline. Those are our sensors to make sure we have the right dose. Those are specimens we put in the beamline, And then we basically reproduce space radiation on the ground. That's my team actually putting some specimen a few years ago. So that's how we do analog radiation on the ground. Physics, so we do a lot of fluids in space, and here you're going to see Scott Kelly, who's actually going to put some dye in a water bubble, and actually he's going to do something. It's one of a kind experiment. Here he puts an Alka-Seltzer inside the bubble and see how the Alka-Seltzer bubble inside in microgravity. And all the physics equations are not so well known, so there's still a lot of physics being done here, and it's really exciting. So it's a great visual. Okay. Last but not least, modeling and artificial intelligence. We spend a lot of time doing AI. This is actually some recent study from Keith Thu, who is actually a PI in the UK. And this is a picture of the kidneys of mice that were in space. And we're going to dive into these kidneys, and what we discover is that there is some disruption of the kidneys. And he's using some very nifty tools here. It's actually a reconstruction of the inside of the kidneys, and you can actually visualize the damage that took place from the ionizing radiation and the space. So now, where do you put all this data? This is where we come in. Open Science Data Repository. We have two databases, one called GeneLab, which focuses on molecular data, so RNA sequencing, DNA sequencing, epigenetic. And then you have ALSDA, which is a database that is physiological data, phenotypic data. We also have a biospecimen archive system that actually I'm going to cover first, where we basically store specimen from experiment on the space station. So here you at NASA Ames Research Center, about 35 miles south from here. This is where we are located. Those are our freezers. You can request this tissue through our database, and you can do experiments that have not been done yet on the leftover tissue. And basically, you have access to specimen and metadata through our database, so you can search it. I will give you the QR code at the end. And then we really cover every single tissue we can have from the mice, and also now we have a collection for microbes. So if you're interested in microbial information, we also have this kind of data at Ames. Next, we do DNA sequencing. This is the Omics database, GeneLab, and one of the special things about our database is we generate our own omics. So that's actually a liquid handler right now, doing the library preparation for sequencing. And then this is the iSeq, which is we typically verify the quality of our DNA first on a small sequencer. This is Valerie Boyko. She's our lead for the sample processing lab. Once she verifies the DNA, she goes and uses those flow cells. In those flow cells, you can do 24 human full genome in 48 hours through the Illumina Nova 6000. There's a new machine that's 2.5 times faster than that now. So we are generating a lot of data very fast. So that's omics. Now let's talk about non-omics, physiological, phenotypic data. So this is much more complex in a way. We're dealing with images, time-lapse, and then microscope images. And the microscope images open a full can of worms because you can look at multiple dimension, X, Y, Z, but also time dependence, multiple channel, histopathology. And we have developed configuration to take any of this data in our system. So even movies, behavioral movies are being taken by us. Again, that's the behavior of the flies here. You had the mice just before. So all this is actually stored into ALSDA, our second database. Okay, how do you access our system? You go to the submission portal, you can click on a study overview, and then here what you're going to do is you get a DOI, which is important. All our data have a DOI, so when you give your data, you can actually, this is actually a visual of the submission portal, so you can actually here look at the sample information. It's a space flight, the type of factors. Let's see, you can actually go back and move to the assays. That experiment's got 4 assays. You can enter as many assays as you want. And for each assay, we have configuration that have been designed with the community. And you can actually click on them and see the actual details of this assay. So I encourage you to play with our database. The other thing about the repo is you can actually visualize the information. So you go to data repo, and then here you can see the different icons for different things. So mice, plants, that's a microbiome experiment on plants. You can search by subject and biospecimen. Let me move. And then, or you can just type the search key. So let's type liver. Obviously, a bunch of mice data comes along. We can click on one of them. You have a quick description of the experiment. You can see the factors here, which are spaceflight. And then you can actually look at the sample level. This is each individual sample level. And then this is the assay table. You can actually download this information. We've been so good at designing those things that we're working with SpaceX on the Inspiration 4, where we're taking both GeneLab data, omics and non-omics data, so whole blood count, So stay tuned, it's coming up in January. We have a big package in Nature coming out on this. You can also visualize the data. So this is classic things such as, this is RNA sequencing transcriptomics, so PCA plot, volcano plot. You can do a pair plot cover. This is if you want to compare two different signals. That's a heat map with the gene expression level. You can download the full change, the p-value. This is a pathway analysis, so you can see how a gene interacts with other genes in a pathway. Those are enriched pathways. So all this is at your fingertip. You can just touch, click, and you are there. Last but not least, on our visual is the environmental data platform. So you can call an experiment. So here I'm looking at temperature, humidity, and CO2 level, radiation level, and I can compare different experiments. So I can compare 3 experiments taking place at different time, and you can see how the dose here is changing as a function of emission. You can download those data and play with them further, but this is really important for the PI when they have an experiment. They can go to the EDA and download the telemetry for their experiment, so very useful. Okay, the community, how do we work with you guys? Well, we have a program called GeneLab for University and GeneLab for High Schools where we train people with our pipeline, bioinformatics. We also work with educators. So we've been focusing on a new model recently where we train the trainers. So we've been focusing on MSI and HBCU. We train them and they train their students. We're also working with other countries. And that program is available to you. They look like notebook. So that's a classic notebook for doing the same thing I just showed you, PCA plot, volcano plot, and so on. We also have a community called AWG. If you want to join us, we have 600 members. We work together. We meet monthly. We define the standards for our community. This is our annual workshop where we strategize about, how we're going to turn the data into something useful. How do we improve our visualization tools? And those are the publications we come up together. So that's Cell paper, Nature paper. We have generated 60 papers today in the big journals through this community. So I'm sorry, it was a bit of a marathon. There's a lot of things to cover, but I want to finish with this beautiful picture of the future. NASA wants to go on the moon and Mars, and something that's often forgotten. We are laser focused on the hardware to go to the moon and Mars, but think about the human factor. To make humans live on these planets, Mars, and to go to the moon, you need to understand the fundamental response of this environment to humans. So we are the essential ingredient in SMD to make this happen. So next time you think about Mars and the great thing we're going to be achieving there, think that it can only happen with the biological and physical science division. So I'll leave you on these pictures and I'll be happy to take any questions. Thank you.